Good evening. Whoa. Oh. Welcome to the Secret Science Club.
Thanks. Thank you. So as Dorian and Margaret said, I work for the Albert and Mary Lasker Foundation. We believe that great science needs public support in order to thrive. And we're really honored to sponsor tonight's speaker at this great venue. Thank you, Bell House. So one of the things I love about my job is meeting fantastic scientists like tonight's speaker, Dr. Jeffrey Friedman. Dr. Friedman is a professor at Rockefeller University, and he's an investigator at the Howard Hughes Medical Institute, some Rockefeller people. In his lab, as you'll hear about tonight, he studies the molecular biology of body weight and food intake. I'd like to say he's also an excellent writer, and he's got a deep appreciation of the history of biological research. There are copies in the back outside of Nature Medicine from 2010, when Jeff won his Lasker Award, in which he wrote a wonderful and moving history of Israel Kleiner. Israel Kleiner was a pioneering diabetes researcher working about 100 years ago, and he almost discovered the insulin hormone. If all the copies have disappeared from the back, you can still find that essay on our website, laskerfoundation.org. So please give a warm welcome to Dr. Jeffrey Friedman. Jeff.
I can hear you. I can't see you. Well, it's really a pleasure to be here, and a bit humbling, actually, to be representing the Lasker Foundation. The Lasker Foundation has and continues to make an enormous impact on biomedical research, and Mary Lasker, in particular, has just made inestimable contributions to the biomedical research enterprise. So I'm going to tell you tonight about our work. Did anyone besides me notice that they were playing Fat Bottom Girls? I was really nervous they were going to play Rockefeller Skank by Fatboy Slim. And so I was probably the only one who was a little relieved to be hearing that. All are true, I suppose. Okay, so I'm going to talk about a subject today that is a little more challenging to talk to the general public about than many others, obesity, and that's because everybody has an opinion, a deep personal experience with their own body weight, the weight of their families, an intimate set of experiences with food that lead each person to develop their own theories and hypotheses about what causes the problem. As Jeff, one of the people who helped me set up, pointed out, if I were here talking about the Large Hadron Collider, it's unlikely anyone would be jumping up and down saying, "No way, you're looking in the wrong window." The experience I have talking about this subject is a little bit different, and so I'm hoping you're willing to indulge me for the next period of time and at least suspend whatever your preconceived ideas are and at least consider the possibility that modern science has something more to tell us about this subject than, frankly, you'll often read in the newspaper. So the subject today is entitled Leptin and the Biologic Basis of Obesity. And in addition to giving a discourse on what we've learned in the last 20 years about this subject, I'm going to do something I've never done before in a presentation to the lay public, which is to try to tell you a research vignette at the end that is absolutely no different than the research vignette I would talk about to a scientific audience. And I'm doing this for a few reasons. First of all, I hope you find it interesting. It reveals an incredibly exciting new set of technology. I think it's pertinent to the topic, and it's pertinent to the topic at hand. So I'll tell you a brief story at the end about leptin and the reward value or the pleasure derived from food. Okay, the subject for tonight. Why is the individual on the left obese and the individual depicted on the right not obese? Now, if you asked people why the difference, everyone would say, well, the individual on the left probably eats too much. And that would be true. But simply saying that ignores the deeper question, which is, why does the individual on the left eat too much? And if nothing else, I think I can explain to you why the individual on the left in this image, in the left image, does eat too much. So what I'd like to do is deconstruct obesity and ask you to take a brief sojourn with me along a road that science would take to approach this problem. And so what we would do for any other medical problem is, first of all, define it and measure its dimensions. We'd want to understand what causes the problem, and with that information, the hope would be that we could intervene in a meaningful way. So how do we measure obesity? Well, obesity technically means an increase in body fat. The normal amount of body fat is 10 to 20% of your weight is body fat. If it's increased, you're said to be obese. There are ways to measure body fat directly. They actually make use of the Archimedes principle in some interesting ways. But without going into that, I'll tell you that in every epidemiologic study that's published on this subject, a surrogate for percent fat is used that some of you may have heard about. It's called body mass index, which is weight in kilograms over height metric in meters squared. And this image here depicts what your BMI would be if you were a 5'10" man and weighed the various amounts there. And what this simply does is say it sort of corrects your weight for how big you are. And generally, if your BMI is higher, it means you have more fat, although frankly, the measure can create difficulties because you'll also have a high BMI if you're muscular. Now, a BMI above 25 is associated with an increased risk of some health problems, but a BMI of 30 is associated not only with an increased risk of health problems, but if extreme enough, an increased risk of mortality. And this is because, for reasons that we don't understand, increased fat mass is associated with diabetes, hypertension, heart disease, a condition known as fatty liver, that can cause cirrhosis, and even some forms of cancer. And these are the principal causes of morbidity and mortality in the developed world and growing problems in the developing world. And the consequence of this is that obese people, or morbidly obese people, don't live as long as leaner people. And that's shown here in a prospective study of 2 million nurses. And just look at the magenta line. What it shows is as BMI starts to go above 30, the relative risk of death goes up. So this is an important public health problem. Now, at some level, we all ask ourselves the question when we encounter an obese person, well, if they're up here and suffer health consequences, and moreover, a social stigma that lead obese people to make less money and be promoted less readily than their no better qualified lean counterparts, why don't people just lose the weight and come down to the flat part of the curve where their health will improve and the stigma will be alleviated? Now, there are three general explanations for why it is people who are overweight and suffer health consequences have difficulty losing the weight or become obese in the first place. And I've listed them here. The first would be to suggest that the obese lack a level of willpower that's normally evident in lean people. I find this point of view is more often favored by lean people. A second possibility is lifestyle and environment. We live in a modern environment where we have greater access to calories, we don't need to move around as much, and that's what causes the problem. And then the third possibility is biology and genes. Now, I want to start by taking a quick vote where everybody can vote their preference. Hopefully I can see you. I'll point out, though, that I used to take a vote before and after I finished. I stopped doing that when fewer people voted for biology when I was done than when I began. So I'm not going to do that. So who thinks this is a problem of willpower? Is it a very politically correct crowd? Actually, who thinks it's lifestyle and environment? A lot of people. And who thinks it's biology and genes? So this is a pretty, well, I'm going to come to that. Okay, we have a vote for all three, which is actually the right answer. But I should digress for a moment and tell you that this was pretty much the vote I normally get. No one ever votes for willpower because it's pretty clear that's not what I don't want to hear. The exception is Canada. In Canada, everybody votes for biology. Of course, I have a feeling if I went to Canada and said, this is a willpower problem, everyone might have voted for willpower. Canadians being so polite. I hope there are no offended Canadians here. I would hate to have been the first person to make a Canadian angry. All right. In any case, the correct answer for this and any other medical condition is it's all three. If we were talking about heart disease or cancer, we would say, well, clearly there's a biological and genetic basis, but lifestyle environment can play a role, and in some cases, personal behavior is relevant. And so the challenge for us, in a way, is not simply to understand how biology and genes might contribute to this problem. The challenge is to figure out not only that, but how environmental and behavioral factors might interplay with the genetic propensities that we each carry. And that's, in a sense, what our ultimate objective is. Now, what I'd like to do for a few moments now is at least go through my view on the relative merits of the cases for each of these three. And then you can draw your own conclusions at the end. And in the question session, don't be at all inhibited about saying whatever you think. Believe me, I've heard it before. Okay, willpower. Well, in a sense, the case for willpower is made constantly in the form of advertisements and infomercials. And to sort of give you a feel for this, I'm going to put up my version of such an infomercial now. Oh, sorry.
Are you tired of being overweight? Tired of the latest weight loss fads? Wouldn't you like a plan that just works? Well, now it's easy. Just follow the Can't Miss Weight Loss program and you too can be slim and trim. Just follow our simple meal plans and your extra pounds will disappear like magic. If you really want to lose weight, decide now to follow the Can't Miss Weight Loss program.
Who's seen an infomercial like that? Yeah. I mean, they're all over the place. If you're up late at night, as I often am, I mean, you can't miss them. What you'd probably also notice was some verbiage scrolling by very rapidly at the bottom. It turns out the FDA now requires diet plans to report their success rates. And if you look carefully at all of the data for all of the different diet plans, it turns out that most diets fail over at least the medium term of 1 to 2 years. And I'm not talking about people who've lost 10 or 15 pounds. People who've lost substantial amounts of weight, say more than 25 or 50 pounds, in general return to the weight they started back with, originally. And so the effectiveness of diets or willpower, as said in the advertisement, decide now, can be seen over the short term, but over the long term, the potency or the effectiveness of this goes down. And there's a pretty good reason for that. Living organisms obey the laws of thermodynamics. So what this means that energy into a system has to be balanced by energy out of the system, or else the amount of stored energy will change. Now, in humans, if you fill in the blanks on this equation, energy in is our food intake, energy out is the calories we consume, and any difference or imbalance between the two will lead to more energy being stored, and in humans, energy is stored as fat. Okay, so how well does the law of thermodynamics explain the fact that body weight is very, very stable in people who are not otherwise trying to change their weight or don't have an intercurrent illness? And this has been evaluated in Sweden, in South Africa, in Israel. Weight in people who are not actively trying to change their weight is stable. Well, how could that be? Over the course, for example, of a decade, we'll eat vast quantities of fat, protein, and carbohydrate, convert that to waste, heat, and a small amount of work, all the while maintaining a relatively constant weight. Now, there is a tendency to put on about 10 pounds per decade. So if you simply do a back-of-the-envelope calculation for this amount of input and a weight change of 10 pounds per decade, what you find is that there must have been a very precise balancing of energy in to energy out that operated with about 99.6% precision or higher. What this means is that quite naturally your body simply counted calories with sufficient precision to maintain your weight despite consuming tons worth of nutrient. I'll point out that this level of precision exceeds the error rates on the calorie counts of the food you eat by several logs. So if you do calorimetry on foods, these are the published error rates. And what I find quite amusing about this is that the more organic the food, the higher the error rate. And we could talk about why that would be. Now, this line of reasoning has suggested for actually more than a century that there must be a biological system that counts calories for us. There must be some biological drive that manages these vast quantities of nutrient flux. And it's very easy to demonstrate this in animals. So in an experiment of this sort, which has been done multiple times, you take two groups of rats, and one you let eat stably, and the other group of rats has a tube put into their throat and is force-fed, sort of like a duck being made into foie gras. Now, if you do this, of course, by thermodynamics, you're introducing more energy into the system. The animal will eat less. But during that interval, its voluntary intake will go down. It's getting all the calories it needs through the tube, so voluntary intake goes down. But then look what happens when you stop force-feeding the animal. It continues to eat less for several weeks or months until it finally approaches the weight of a control group and ultimately merges with it. It's as if somehow the animal knows that it had been made too fat and elicits a biological response to return its weight to some predetermined set point. Similarly, if you food restrict an animal for a group of animals for a period of time, of course they'll lose weight because of the law of thermodynamics, but as soon as you give that animal free access to food again, its weight returns precisely to that of a control group, as if there's a biological drive that seeks to restore the weight of that animal to some predetermined set point. And the presumption is that the same basic drive is active in humans, and this explains why the success rate of dieting is as low as it turns out to be. So I think it's very clear that willpower can work over the short term, but the available evidence suggests that some other basic drive intervenes over the long term and acts to return weight to a predetermined level. And we'll talk about what the physiologic mechanisms that account for this are in a moment. But first I want to go through the case for lifestyle and environment. Well, that case is made all the time. You might have seen quotes of this sort: no age has afforded more instances of corpulency than our own. This was Thomas Short in 1727. I have no idea who Thomas Short was, other than he was short. All right, anyway, I'll also point out to you that the oldest sculpture ever identified, the Venus of Willendorf, was not exactly trim, and clearly there were very obese people throughout the ages, documented in the Pickwick Papers and in this cartoon here. So why the disconnect? What is the nature of the problem today as compared to previous years? Well, the sort of case for a major shift in obesity in recent decades is made based on the following sorts of analyses. Basically, you look at the number of people who exceeded this threshold of BMI over 30 some period of time ago. You look at it again today, and what you would see if you analyze the data in this way is a doubling of the obesity rate between 1980 and 2000. And this is the source of much of the, to some extent, appropriate alarm or concern about ways to remedy this problem, especially given that this has health consequences. Moreover, people look at this data and then say, well, our genes haven't changed during that period, so it has to be the environment. First of all, I'll just digress for a moment and tell you that very prominent genetic shifts can occur in populations over a short period of time. I don't know if any of you have read The Beak of the Finch. It's a wonderful Pulitzer Prize-winning book by John Wiener, And it outlines the work of some Princeton scientists who show that you can have very dramatic genetic shifts in response to environmental changes in finch populations over the short term. But setting that aside, what's the case for this argument? Does this sort of observation refute the idea that this medical problem has a genetic underpinning? Well, I would argue that data of the sort that I just presented gives you only a partial understanding of what's going on. And it has to do with the fact that BMI is a distributed trait. It goes from low to high and is sort of distributed along a more or less a bell-shaped curve. At the same time, we measure obesity as a dichotomous trait, meaning if your BMI is over 30, you're obese. If it's under 30, you're not obese, you're overweight, and so on. Over 25, you're overweight. Under 25, you're not. Now, there's a problem in epidemiology when you define a continuous variable with a fixed threshold. And let me show you what I mean by this, looking now at the data for obesity rates between 1990 and 2000. Okay, the threshold is BMI over 30. This is a schematic curve. On the next slide, I'll show you a real curve in a moment. The average BMI in 1990 was 26.7, and 23.3% of the population was obese. In 2000, the average BMI was what, 28.1, and now 30% of the population is obese. So what happens is if you have a distributed trait, the curve doesn't have to move very far for you to get a marked increase in the number of people who exceed the threshold. So in the 1990s, for example, there was a 33% increase in the obesity rate, but this was associated with a 1.4 average increase in BMI, or 7 to 10 pounds. And so what I would argue is you get a very different impression of what's happening if you hear obesity rates go up by a third in a single decade as compared to hearing average body weight went up 7 to 10 pounds. Moreover, the data that are generated and generally published don't typically take into account or correct for age and sex. And if you do so for men and women at a variety of ages, I'm only showing you ages 40 to 59 now, it turns out the curves don't look anywhere near as different as you would imagine they would when you hear obesity rate went up by a third in a single decade. Now, I'm not here to argue against the importance of environment, I'm here to tell you that this is an important shift. There are clearly more obese people now than there were then, and if we could reduce this average BMI increase, that would be really good for public health. So we certainly want to understand what in our environment is responsible for this increase, and we could talk about this a little bit later, although my view is that the environment is a necessary but not sufficient condition for the development of obesity. That said, there's another question embedded in data of this sort, which is this. If we look at a cross-section of our population today, in an environment where everyone has essentially unlimited access to calories, weight is incredibly variable. So how is it that in today's environment, where we can all pretty much consume as many or as few calories as we choose to, some people might weigh over 400 pounds and other people less than 100 pounds? And the answer to this specific question is, to a very great extent, genes. Now, there are a number of ways to assess the genetic contribution to a human trait. I won't go through all of them, but the data are consistent. The most quantitative data come from systematic analyses of twins. And what you can see here are twin pairs, the body habitus of twin pairs. These are monozygotic and these are dizygotic twins. If monozygotic twins are more alike for a trait than dizygous or fraternal twins, we can assess and even quantitate the extent of the genetic contribution of that trait. And it turns out, if you do a systematic analysis of this sort, obesity turns out to be as or more heritable as any trait that's been studied, with the exception of height. So 80 to 90% of the variance in the rates of obesity can be accounted for by genetic factors. Higher heritability than schizophrenia, diabetes, hypertension, alcoholism, heart disease, all of which people generally accept have a genetic basis. Now, to give you a sense of what these genetic factors might be, I want to tell you about a case report. And the case report is about the child whose image I began by showing you. This child was characterized or first brought to attention by Stephen O'Reilly and colleagues at Cambridge, England. The child was of normal weight at birth, but began to develop morbid obesity in infancy. He showed marked hyperphagia, which we'll talk about later. He was already pre-diabetic at the age of 4 when he weighed 90 pounds with 57% body fat. Now, this child had a similarly affected 8-year-old cousin who weighed 200 pounds, which is what I weigh. All right, 210 pounds. Well, actually, it's 215. It is 215. Now, these children came from a highly inbred pedigree, and it's often the case that when you have an inbred pedigree with so-called consanguineous marriage, that often reveals single gene or recessive disorders that cause that condition. And O'Reilly and colleagues noticed some important similarities between the characteristics of this child and his first cousin and a strain of mouse that we'd been studying. This is a picture of the OB mouse. It was first reported by George Snell at the Jackson Laboratory in Maine. Snell won a Nobel Prize for breeding mice to show how immune tolerance was controlled in animals. Now, in the course of those inbreeding experiments, similar to what I described for human, they noticed one day, I was told by Skippy Lane, Snell's technician, a mouse that looked a little bit plump at weaning. And at first they thought the mouse was pregnant, except for 3 factors. First, the mouse kept getting fatter. Second, the mouse never delivered. And third, the mouse was male. This is true. She told me this. You could see why Snell won a Nobel Prize, I think. In any case, by all genetic criteria, the only difference between this animal and his sibling littermate is a defect in a single gene. And the result of that genetic defect is an animal that eats voraciously, weighs 3 times a normal mouse, has 5 times as much fat, despite the fact that both animals have the same free access to calories. Now, my laboratory in 1994 identified the defective gene in these animals as encoding a novel hormone which we named leptin. Leptin is a hormone. So hormones, as you probably know, are molecules made by one organ in the body that circulate in blood and act on another organ. Leptin is a hormone made by fat tissue in proportion to its mass. It circulates in the blood and then acts on brain centers in a region known as the hypothalamus to control appetite. And it's what's referred to as the afferent signal in a feedback loop that regulates body weight, similar to the way that, say, temperature is the afferent signal in a feedback loop by which a thermostat controls temperature. So leptin is made when you have more fat, and then it acts on the brain to suppress appetite. And the problem in the OB mouse was that there was a mutation in the gene, so fat tissue no longer made leptin. So this animal was fat because it had no leptin in the blood. And so in this image, what we show is what happens if you give an OB mouse saline. Not much, it's still pretty fat, and you give it leptin, it loses weight. What I find most remarkable about this slide are the perfectly parallel tails. The graduate student I work with on this, Jeff Hollis, in fact, it was about the 4th time I showed this slide. I looked at it and I went a little cross-eyed. I said, what happened here? And I asked Hollis how it could be the tails are perfectly parallel. I thought he might have glued them, but he denies this, which of course he would. Now, in addition to reducing food intake to account for this, we noticed something really interesting when we videoed the animal, which is, and this is the first thing you see if you give an animal leptin made in the laboratory, I should say. We did this with Stephen Burley. We could make the leptin in a test tube, and if you give it now, the OB mouse not given saline just sits in the corner, not moving around much, but the OB mouse given leptin moves around normally. I'd like to point out that we generally think activity is under voluntary control, much the same as food intake, but this begins to suggest that our overall activity level may be set by biological factors, not simply based on the decision about whether or not we think we ought to exercise. Okay, so these data established that leptin was a hormone that regulated weight, and based on this work, O'Reilly went ahead and tested the leptin levels in the child I showed you a picture of. And it turns out he and his cousin made no leptin whatsoever. So they gave the child leptin, made in the laboratory, and here's what happened. The child got older. This has nothing to do with leptin. That's just the way things are. Despite getting taller, the child lost weight with the reduced body fat, and he became less diabetic shortly thereafter. This was also done by Steve O'Reilly and his collaborator, Sadaf Faruqi. And now here's what the child looks like after 5 years of leptin. These were the two images I began by showing you a picture of at the beginning of my presentation. Same child. So in this child, we know that the problem was not a lack of willpower. It was not a lifestyle, a set of poor lifestyle choices. Rather, in this case, this child lacked the hormone leptin. Now, leptin mutations as a cause of obesity are very, very rare. That's true generally about hormones. They're poor targets for mutations. There are only a few dozen such cases. But as I'll come to in a minute, it turns out a very substantial proportion of morbid obesity is associated with mutations in other genes in the pathway on which leptin acts. We'll come to that in a minute, but first I'll just point out that what happens in this child is he fails to make leptin. So his brain interprets that as meaning there's no available fat tissue, because generally the more fat, the more leptin, the less fat, the less leptin. So in the absence of leptin, this child's brain thinks he's starving. And the normal response to starvation is to eat more, fat mass expands, but it never makes the signal that shuts the system off. And so based on this and a host of other work, the following, let me back up here. The following mechanism has been invoked, and I think this is supported by a great deal of experimental evidence. What's going on here? Okay, at our stable weight, we have a certain amount of fat, and our food intake equals our energy expenditure. If we lose weight by dieting or because of a mutation in the leptin gene, leptin level is low. This provides a stimulus to eat more. You'll see that in a moment. So you've lost weight, leptin level goes down, and this will provide a stimulus to eat more until such time as your fat mass and leptin level return to the starting point. Conversely, if you go on a binge of eating and you're normally leptin responsive, fat mass and leptin level will go up, and this will suppress appetite again until fat mass and leptin level return to the starting point. And so this sort of begins to explain the animal data I showed you earlier about how animals seem to be able to find their way to some stable level for weight. Now there's an important point here, which is that leptin isn't the only factor that regulates weight. I'll come back to this, but it nonetheless is a factor. And it means that if you want to change your weight in either direction, there's a biological force that resists that change. So if you want to either increase or decrease your weight, you'll have to overcome the biological force encoded by the changes in leptin level. Now, this, I think, has important implications for people who are dieting, because it means if you're going to maintain your weight loss on a diet, you're going to have to overcome the really powerful biological signal that's being conveyed by a low leptin level for as long as you want to keep that weight off. Okay, now with that, I want to digress for a moment and tell you that if you see a robust, what's referred to as homeostatic system of this sort, where some biological parameter is kept within a relatively narrow range, it usually has evolutionary implications. And it's worth, I think, therefore, talking about what are the evolutionary considerations that would lead biological systems to stably maintain their weight within a narrow range. Now, this list is not exhaustive, but it gives you a feel for some of the factors. Our forebears, hunter-gatherers, would have faced risks if they were either too lean or too obese. If you're too lean, you're at greater risk for starvation the next time a famine comes around, so you want to have enough fat to provide a source of energy when food is no longer available. But if you're too obese, you're at increased risk of being eaten by predators. And actually, this is true. It's been shown in bird species, and if you look at fossil records from the savannah in Africa, it turns out a lot of our forebears died as a result of attacks by predators, because obese individuals are less able to escape in many cases. And by this I mean morbidly obese people. There may have been other selections, but this gives you a feel for the fact that at least in a wild environment, there are powerful selective pressures that would tend to want to balance out the relative risks of either extreme and put most of individuals in the middle. Now, different populations today differ with respect to their propensity of obesity, and to some extent this may reflect what the relative risks of these two possibilities were in the environment in which that population used to live. Okay, well, what I told you about so far is that leptin is a hormone that regulates weight. I also told you that leptin deficiency as a result of mutations can cause massive obesity. I won't tell you about other implications of this research for other human disorders, but there are other low leptin states, and leptin is a very effective treatment for many of these. In fact, leptin has been approved for the treatment of this form of diabetes known as lipodystrophy in Japan, and it's being reviewed by the FDA now for possible approval in the United States as well as approval in Europe. I'm not going to talk about this, however, because today's topic is obesity. And what I want to tell you now is that obesity is not a leptin deficiency syndrome in anything other than the very few number of cases where patients have a mutation in the leptin gene. If you look at obese patients, what you find is the more obese they are, the higher, not the lower, the leptin level is. Now, when you see an individual with the high levels of a hormone, in the absence of the hormone effect, in this case to reduce food intake and reduce body weight, it usually means that this is a hormone resistance syndrome. And what that means is you make the hormone perfectly well, but for one reason or another, the hormone you make doesn't work well enough at its target organ. And the classic example of a hormone resistance syndrome is type 2 diabetes. 90% of diabetics actually overproduce insulin, but they're diabetic because the insulin they overproduce doesn't work well enough. We still don't understand why that is, in fact. The same, however, appears to be the case for leptin. That is, most human obesity is associated with high leptin levels and an apparent resistance to leptin. What this means is that somehow there's a relative block to leptin's action at its site of action in the brain. So less effective signal gets through, you'll eat more, burn less energy, fat mass will expand, as if by dialing down leptin sensitivity, you'll dial up fat mass and leptin level. So you're resetting weight higher the less sensitive you are to leptin. And all the available evidence now suggests that obesity is to a large extent a result of a resistance to the endogenous leptin you make. Now, the empiric question that devolves from this is, well, okay, what happens if you give obese patients extra leptin? Well, maybe something would happen. On the other hand, maybe nothing would happen because they're already making a great deal of their own version of the hormone. Well, the earliest experiments giving leptin to obese patients were in fact quite encouraging. And what's shown here are admittedly a small number of patients in green getting leptin and in red getting a control solution. And what was seen was actually a pretty significant weight loss in some, but not all of the patients, at a dose here of 0.3 milligrams per kilogram of leptin. I mentioned the dose for an important reason, though, which is that this was a really high dose. This required that patients give themselves two milliliters injections a day. And the company that was testing it at the time correctly concluded that there's just no way that this could be used as, in a clinical setting with any great frequency. And so after a lot of additional experiments, they decided that they were not going to pursue leptin as a treatment for obesity any longer, although, as I mentioned, it does have efficacy for a number of other conditions. So at that point, research into leptin as a therapeutic foundered, but there are a number of things one could have imagined doing. Well, one possibility is that some patients do respond better to leptin than other patients, maybe even lower levels. It turns out a significant subset of obese patients do lose a substantial amount of weight after leptin treatment. And there's reason to think that responder fraction might be that subset of patients who happen to make lower leptin levels to begin with. There's another approach, which would be, well, maybe we can combine leptin with other agents that might amplify its effects, and we would call such things a leptin sensitizer. And what I'm going to tell you now is that a leptin sensitizer has been identified, and that perhaps the use of leptin in combination with this other agent might reveal some efficacy that hasn't been seen with leptin alone. Now, to tell you that story, I need to remind you of something I said earlier, which is that leptin's not the only signal that regulates weight or food intake. Food intake is thought to be regulated by a short-term system and a long-term system. The long-term system's job is to maintain relatively constant levels of body fat, and leptin is one of the key components of the long-term system. The job of the short-term system is to keep the nutrient levels in your blood relatively constant during the day. And so the short-term system controls how hungry you get between meals and how full you get during a meal. And the signals in the short-term system are not things like leptin or its components. Rather, they're a set of hormones and neural signals from the gut that monitor how much food you've eaten at a given meal. And some of you may have heard the names of some of these short-term signals. Ghrelin has received a lot of attention recently, cholecystokinin, peptide YY, and many, many others, as well as I mentioned, neural signals from gut to brain. Now, one of the signals here, amylin, is another hormone made from pancreas. And amylin, it turns out, is used as an anti-diabetic therapy. It's not commonly used, but amylin is an anti-diabetic therapy, and it actually also can induce a small amount of weight loss in the similar range to what's been reported for leptin. Now, many people have argued that if you have a short and a long-term system that talk to each other, you're probably going to need a combination of therapies that target both. Because if you target just the short-term system, the long-term system will compensate and vice versa. And so the idea has been out there for a while that we may need to combine short-term and long-term signals. And so a company that no longer exists, Amylin Pharmaceuticals, they've been acquired by Bristol Myers, decided to test a combination of leptin plus amylin, a long-term signal and a short-term signal. And now they saw something that wasn't seen with either compound alone. Now the combination led to about 14% weight loss in the treatment group. And this was a level of efficacy that's considerable and higher than that for any other FDA or registration studies it's known to that I know of. So this at least raises the promise that in time leptin might be combined with other agents to achieve more effective weight loss that can be achieved with leptin alone. Well, that's it for the empiric part of the talk. I want to now turn to the basic science we and others have done to try to understand more about how leptin works and what the causes of leptin resistance might be. As I mentioned before, leptin acts on nerve cells that control appetite. Presumably, in the leptin-resistant state, it doesn't work as well on the population of nerve cells that it effectively works on in lean patients. So there's some block to leptin action in terms of its ability to modulate neural activity. And as I mentioned, and we'll show you now, the neurons that primarily respond to leptin are in a part of the brain known as the hypothalamus. The hypothalamus is a part of the bottom of your brain that controls all basic behaviors to a large extent. Food intake, body temperature, sexual behavior. Why is this not on? Here we go. Sexual behavior and so on. In addition, the hypothalamus is pretty much unchanged between fish and humans. You can recognize all the structural features of a fish hypothalamus as not being too different from a human hypothalamus. So what that slide just showed is a section of the brain, and the hypothalamus is at the base of the brain, and leptin acts on these two nuclei here. Nuclei are collections of nerve cells known as the arcuate nucleus and the VMH, or ventromedial hypothalamic nucleus. And we've even learned in recent years the identity of several of the key responding neural populations. And we now know that the way leptin works is by inhibiting neurons that activate appetite and activating neurons that inhibit appetite. Now, in green is one such neural population. It expresses the receptor for leptin. A receptor is sort of like the receiver for a hormonal signal. A radio wave emits a radio signal. The receiver receives and translates that signal. Same thing with the hormone receptor. And there's a class of neurons in the hypothalamus that express this receptor and a peptide called neuropeptide Y. And when these neurons fire, an animal or a human will eat more. A second population expresses the leptin receptor, but now a different peptide known as MSH, and when these neurons fire, they activate a different receptor called the MC4 receptor, and animals or humans will eat less. Leptin works by inhibiting these green neurons and activating these red neurons. So if you make no leptin, the green neurons are maximally active and the red neurons are off and you'll eat more. If you give extra leptin, red becomes more active, green becomes inactive and you eat less. And the system is said to be tuned, which means that quantitative changes in the leptin level changes the relative activity of each of these two pathways. Now, one interesting feature of this wiring diagram is that it actually isn't so different from the general wiring diagram that drives food intake in a worm. In worms, there are pairs of neurons that either activate or inhibit food intake, and they receive external cues that drive behavior in one direction or another. Same is the case in animals and humans, where these neurons sense leptin as well as other signals, including some of the short-term signals I mentioned. Now, the reason these pathways have attracted a great deal of attention in the field in which I work in recent years is because mutations in it cause human obesity. And this includes not just leptin, but the leptin receptor, alpha MSH, and the MC4 receptor. And we now know that probably close to 15% of morbid human obesity is caused by a mutation in one of these 4 genes or 1 or 2 others that I didn't put on this diagram. So that means if you see someone morbidly obese on the street, there's a 15% chance that they'll have a mutation in a gene the identity of which we already know. Moreover, my strong suspicion is that with the advent of whole genome sequencing, which I suspect most of you have heard about, we'll identify more and more genes over time that contribute to the development of obesity. In general, the more obese someone is, the more likely it is to be genetic, and we and others are now conducting studies that use DNA sequencing to try to identify these other genes, all of which to date, in fact, map on top of this neural circuit. I'll also point out to you that these are single gene disorders, and the level of Mendelian or single gene inheritance that causes obesity is higher than for any other trait that's been studied, at least for genes that have been identified. So I think this, in the end, challenges our view that obese people are strictly responsible for their condition. There's at least as much genetic underpinning, perhaps more, to explain the genesis of their condition than there is for any other complex trait under study. Okay, well, that's what I really have to say about the background. I want to now start to turn our attention to the research vignette I threatened to tell you about at the beginning of the talk. And so the story here really is this. We have a hormone, we have a defined number of cells in a specific brain region that respond, and we have a measurable behavioral output in animals, and as I'll show you in human. So what happens in between? How does this signal change a complex behavior? And I'm going to tell you a brief story now about how leptin works by regulating not just the hypothalamus, but another brain region. So as I've told you already, leptin works on the hypothalamus, and as I also alluded to, it's part of a pathway that's referred to oftentimes as homeostatic feeding. It's the feeding that makes you hungry when you haven't eaten a meal, and so on. But this isn't the only part of the brain that can drive feeding. There's a part of the brain that's often referred to as the reward center, and the region is known as the ventral tegmental area, and this brain region plays a role in conveying pleasure, oftentimes in response to palatable food. And so it's also known that sucrose, a sweet sugar, can activate the ventral tegmental area. This region will then produce dopamine, which is released at a variety of places and signals pleasure. And so the question is, to what extent might leptin play a role to regulate hedonic feeding, as it's called, or pleasure-based feeding? Does homeostatic feeding have anything to do with pleasure-based feeding? And that was the set of questions that a very talented postdoc, Ana Domingos, in the lab took on. Ana's now a faculty member back in her home country of Lisbon, and she was also a PhD student of Leslie Vassal, who I know spoke at an earlier Secret Science event. So what we wanted to know was, what is the functional relationship between leptin and the reward value or pleasure derived from food? Now, the tool we're going to use in these experiments is a relatively new and extraordinary one that allows you to very specifically activate defined population of nerve cells using light. Let me explain how this works. It's based on the observation that flagellates, which are single-celled organisms such as Chlamydomonas, move towards light. There's a picture of Chlamydomonas, and if you shine a light, these animals will, or these cells will move in the direction of the light. And a group from Germany, Hagman, Nagel, and Bamberg, showed that this happens because these animals, or these cells, express a channel. And when this particular channel in the membrane of the cell sees blue light, it opens up, sodium flows in, and activates the cell, and that's what accounts to the movement. Now, one of our collaborators years ago, Carl Dieserath, who's justifiably receiving a great deal of positive attention for this, showed that you could take this same chlamydomonas channel, modify it slightly, and now put it into mammalian or frog cells, or frog oocyte cells, and get the same response. And what this shows is if you now expose cells expressing this channel to blue light, currents of sodium flow into the cell. He then went on to put the same channel into nerve cells and showed that you can turn nerve cells on and off just with light. And I'm going to show you that in a second. But first I'll tell you that we wanted to study the role of reward centers in the response to leptin. And this completely dark, unviewable slide, I guess the lights here make that, oh, thank you. Isn't that pretty? So this saddle-shaped structure is the VTA. These are dopamine-producing cells in the brain. These neurons play a role in conveying reward. And what this just shows is that channel has been specifically introduced into this population of nerve cells and none others, which means that in principle, you could shine a light on these cells and they'll release dopamine. Now, here's the way we set up the experiment. In these studies, the animal controls his own reward. The animal licks a bottle of water. The licks can be counted, and every time he licks 5 times, a laser that's placed near these channel rhodopsin, as it's referred to, expressing neurons, fires and turns them on. 5 licks give you a second of light, and here's what happens. Not much. Light comes on, the animal licks, the light comes on, and so on. And this happened. Okay, so what this means in a sense is the animal can now control its own level of reward by licking. It licks, it gets more dopamine, it licks some more. And this is quantitated here. You can see that when the licking is coupled to the light activation of the cells, it licks a lot, and not so much when there's no light. And that's quantitated here. Okay, so we now have a system where the animal can control its own level of dopamine. And what I want to tell you now is how we use this system to show that leptin regulates the pleasure derived from eating sucrose. And to do that, I need to back up and tell you about something called the post-ingestive effect of sucrose. And what the post-ingestive effect of sucrose refers to is the fact that animals and humans prefer sucrose to artificial sweeteners as a result of the fact that they have nutrient value and sucralose, an artificial sweetener, does not. The way the experiment, which was done by Anthony Sclafani at Brooklyn College, works is this. You give an animal a choice where it can lick at either of 2 ports. If it licks at the maroon port here, it gets sucralose delivered into its blood. This is an artificial sweetener that has no nutrient value. If it licks at the yellowish port here, it gets sucrose delivered into the blood. So you set up the experiment like this for a little while, and then you no longer couple the sucrose or sucralose to the ingestion at one or the other port and ask, which port does the animal prefer? What you can see is now there's an overwhelming preference for the side that was associated with sucrose relative to sucralose. This clearly indicates that sucrose is preferred, but it also shows that sucrose is being sensed in the body, not just by taste. And it's the sucrose in the blood that is in part accounts for the preference for this natural sweetener. A few more facts about sucrose and sucralose. Well, the value of sucrose is conveyed, therefore, by a combination of both its sweet taste and this so-called post-ingestive effect, where it's thought the nutrient value of the sucrose is somehow sensed. Now, that actually has been formally tested by another collaborator, Ivan Diarojo, and he showed that it's the nutrient value of the sucrose that's responsible for this post-ingestive preference, and that's because if you add a sugar that can't be metabolized, there's no preferences established. Second of all, even mice that can't taste at all because of a mutation still prefer sucrose to sucralose. So there's some other sensing mechanism for the nutrient in sucrose. And lastly, and this is why we did the experiment we did, sucrose results in an increase in dopamine in these same brain regions, whereas sucralose does not. So the post-ingestive effect is mediated by an increase of dopamine in reward centers. Okay, so what do we have now in our system? Well, sucrose is preferred because it combines sweet taste plus dopamine release, and we can take these two components apart, substituting for sweet taste alone with sucralose, and we can substitute for dopamine release using the laser stimulation protocol I showed you. And so our first objective was, what's the hierarchy of these preferences? And to do that now, we do an experiment in which we give animals a choice. At one port, they get sucrose or sucralose, one or the other, and at the other port, every time they lick, they get dopamine release. So what's the relative preference? So if you look on the left, what we're asking is, which does an animal prefer, sucrose or laser activation of dopamine? The red's a control, so just look at the blue, and what you'll note is, at 0, laser is preferred, but very quickly, as sucrose concentration increases, the preference shifts, and the animals prefer sucrose to laser or dopamine alone. And that's because sucrose has taste and dopamine, the laser only has dopamine. Sucralose is different. Even at the highest concentrations of sucralose, you never exceed the preference for laser. So what this tells you is that sucrose is preferred to laser, is preferred to sucralose, and in a sense what it tells us is that dopamine combined with sweet taste is preferred to dopamine alone, is preferred to sweet taste alone. Okay, so here now, this all is a lead into the experiment. So in these studies, we replicate the components of sucrose by substituting for the sweet taste using sucralose, and the post-ingestive effect by using optogenetics is referred to light stimulation to stimulate dopamine release. And now the question is, what happens if we compare sucrose to sucralose plus laser, a means for combining both modalities? And what happens? Well, now the preference shifts. What's shown here in red is an animal without the laser on. It prefers sucrose to sucralose. But if we now, in the blue here, compare the sucralose with the laser, the preference shifts. And the way we're showing this is that we're just counting the number of licks at the left port versus the right port. Okay, so sucralose plus laser provides a stronger combined signal than sucrose alone. Now this then leads to the experiment I wanted to tell you about, which is what happens to this preference if you fast an animal and change its metabolic state or give it leptin? And the answer is shown here. If you now fast an animal, what you see is the preference shifts again from sucralose plus laser back to sucrose. It's as if the reward value of sucrose has been amplified when the animal has been fasted and starved. So the pleasure it derives from sucrose is being altered by the fact that its metabolic state has changed. Is that leptin? Well, we can test that. We can fast an animal and give it leptin back, and now we shift the preference back again. So leptin reduces the pleasure derived from consuming sucrose. That pleasure is amplified by fasting, and the pleasure or hedonic value, measured in part by dopamine release, is altered by leptin. And so what it tells us is that leptin reduces food intake in part by modulating the relative pleasure that can be derived from food. And that's sort of depicted on this graphic. With low leptin levels in times of starvation, the value of sucrose is amplified, but if you give extra leptin, the value of sucrose is reduced, and this changes our preferences for the combination. So the last bit, is this relevant for human? Well, does leptin play a role in human food intake? Let's return to the leptin-deficient child. Before the first leptin injection, this child ate 2,000 kilocalories a day. Ate 2,000 kilocalories in a single meal. They gave this child a test meal. He ate the whole meal in one setting. I should remind you that 2,000 kilocalories is the same number of calories an adult of my size would eat in a day. All right, 2,500 calories, but after a few leptin injections now, the child ate an age-appropriate number of calories. So leptin regulates food intake in humans, and here again, what's driving food intake in this child is not willpower, is not environment, it's a lack of leptin. Are the studies I just told you in animals relevant for human? Well, The target of dopamine is a region of the brain or reward center known as the nucleus accumbens. The leptin-deficient child shows tonic or constant activation of this dopamine-sensitive region on an fMRI scan. If you give it leptin, you suppress this signal. So the same regions are involved in humans. And lastly, does leptin affect the pleasure that you get from eating food if you're leptin deficient? And the answer is it does. It was interesting. Before the first leptin injection, they asked the leptin deficient patients to state how much they like or how much pleasure they get from different types of food. And what you'll see here is they actually like all foods maximally. That's shown by the bar here with the very, very happy yellow face here and here. And interestingly, they have no preference for cake versus, I think that's cake, for cauliflower. And this sort of makes sense. If you're starving, it really doesn't matter to you so much what food you get. You want any food you can get your hands on. You now give these children leptin, and their preference shifts. Now you see a food preference. They don't like cauliflower anymore, and they like chocolate cake, but not as much as they do before they get leptin. So what this is telling us is that the same mechanisms are likely to be relevant in human also. And let me take a moment or two to tell you what I think this means. When we feel hungry or when we eat, we get pleasure from our food. And we think, probably in part for some Calvinist reason, that if we could simply not partake of that pleasure with willpower, we could change our weight. And what this data says is the pleasure we take is not really an operation that's defining what our level of willpower is. The pleasure we get from eating is a result of sensing of biological signals. So the way we know we're hungry, in part, is not because we can consciously tell what our leptin levels are. The way we tell we're hungry is we get more pleasure from the food we eat. And I think this confusion about what's really driving food intake and behavior in this case is part of what explains the dichotomy or the vast array of opinions about what causes obesity. All right, well, that's the end of the vignette. I just want to close now with a few conclusion slides. First, telling you that there's lots more to be learned. Feeding is what's known as a complex motivational behavior. As I mentioned to you, many factors are relevant to eating, not just leptin level. Other hormones and metabolic signals, sensory factors, vision, smell, taste, emotional factors are important, and in human volition is important, the cognitive wish to either eat more or less. It's assumed based on this that there must be a part of our brain that processes all this information and then makes a behavioral decision about whether or not to eat. Not only do we not understand how complex information of this sort is processed to generate a behavior, we don't even know where that information is processed. So a lot needs to be learned about how behavioral decisions in response to complex stimuli are encoded. Now, despite the fact that we don't know anywhere near as much as we need to about this complex neural circuit, I'll close by telling you that we've learned a lot about the endocrine circuit that regulates weight. It's a classic feedback loop comprised of an afferent signal and leptin, brain centers that receive that signal, and a series of brain outputs that regulate behavior and metabolism in a number of organs. I'd like to believe that this framework will also provide a better means for assessing how environmental and psychological and behavioral factors might influence the activity of this system, and would argue that it's only by a deeper understanding of these mechanisms that effective therapies will emerge. The sort of recommendation the obese get is they should eat less and walk more or exercise more. I'll point out to you that that recommendation was made by Hippocrates 2,000 years ago, and I'd like to think that modern science could do better than repeating a 2,000-year-old recommendation. Rather, I think we should approach this problem the way we would any other medical condition. Define the molecular components of the pathway that regulates weight, understand what's wrong or broken in that pathway in the pathologic state, and as I mentioned, understand how environmental and other factors influence that pathway. And from this will come rational therapies. This was the course that was taken for the war on cancer. It's proving effective now, although it takes time. And this is the way I think progress will ultimately be made. Now, I'm often asked, well, you're telling me this takes time. What should we do in the meantime? And what would I recommend to obese patients? Well, mind you, we're talking about medical therapies here, and what I didn't mention before is that surgical therapies for morbid obesity actually are thought to work by modulating the short-term signals I mentioned earlier, and I think surgical therapy does have a place for very sick patients who are morbidly obese. But what about the rest of the population that's obese? Turns out the advice I would give to the obese are pretty much the same as I would give to anyone in advance of better medical therapies. First of all, focus on improving health, not losing weight. Health is the issue here, and what drives people's behavior in many cases is not their health, but rather the stigma or the perception that, unfortunately, our culture often applies to the obese. So if the focus is on health, what can you do? Well, exercise and eat a hard, healthy diet, which is something everybody should do. In addition, it's known that even modest weight loss can have a disproportionate health benefit. So if you're obese and have an associated medical condition, do your best to lose some weight. As I mentioned, even a modest weight loss can lead to an improvement. I'll also add that there's no evidence that if you're obese and don't suffer health consequences such as diabetes or heart disease, there's no evidence that your health will necessarily improve by losing weight. So I would reserve weight loss for patients who have a health problem. Lastly, don't berate yourself if you cannot normalize your weight. You're fighting against a basic biological system that's very powerful. And finally, don't berate anybody else either. All right, so I want to tell you that I put many of these thoughts into a Newsweek editorial. Some of you remember there was once a magazine called Newsweek. Quaint times they were. And I really liked the tagline. I didn't write this, the editor did, but the editor wrote, "The real cause of obesity, it's not gluttony, it's genetics. Why our moralizing misses the point." And I put in this article many of the ideas that I just presented in the last several hours. And it's been a little under an hour. It turns out this generated a crazy amount of controversy. There were chat rooms on Newsweek, there were chat rooms on Yahoo, and in general, it broke down probably 3-to-1 detractor to supporter of what it is I had to say. So I want to give you a sampling of what people had to say about these ideas, and also to empower you to say whatever you want, because I've heard it all. So I want to share with you what some of the comments were, some of which are actually kind of illuminating. This is crap. I learned reading the chat room, and there were literally hundreds of postings, how many different ways there are to say this is crap. There was, "What a load of apologistic crap," or "ditto to the hogwash." Now, one of my favorite ones was an exchange between a detractor, "I Smell Smoke," and a supporter, Team Hassan. "I Smell Smoke" wrote, "I'll bet Dr. Friedman's buddies don't call him skinny." And my defender said, "And I bet Smoke's buddies don't call him smart." All right. And the last few, I think, actually reveal something about the culture for obese people and why it is this is such a hot-button issue for many people. One person wrote, "I don't think how I live my life is anyone else's business. My obesity is not hurting anyone else." Another person wrote, "I often wonder if people vilify obese people because they're so visibly obvious, and nothing makes you feel better than pointing out the mote in someone else's eye." And the last one I thought was really interesting. Toolkin, no relation to Lord of the Rings guy, I hope. "Are we animals, or do we have an intellect and the ability to choose how we behave?" In a way, I think that's part of the issue. I think humans, as our species, is uncomfortable with the idea that there may be basic drives over which we have limited conscious control. And I think it's worth having a discussion about what the relative potency of a conscious wish would be versus a basic behavioral drive. That dramatic tension plays out for every behavior, and this is one of many examples of that. So with that, I'll stop, and I'd be happy to take any questions you might have. I really appreciate